Hi everyone, my name is Pedro Medeiros, and today I'm going to present to you a little bit of the relevance of bioinformatics for pharmaceutical science. First of all, I'd like to thank Sofia Sueyoshi and IPSF for the invitement for this presentation, and I hope that at the end of this presentation we can have a clear vision of the perspectives for the present and for the future of bioinformatics in pharmaceutical science. So let's get it done, right? Okay. So who am I? My name is Pedro Medeiros. I'm a 34 years old Brazilian bioinformatician. I'm actually first a biologist and then I'm a pharmacist also by the University of São Paulo. And I have a master's in biosystems by the Federal University of ABC, where I develop research on molecular evolution of proteins. I also work in TauGC Bioinformatics as a bioinformatician, a Brazilian company for computational biology and computational genomics. Uh, so check out my LinkedIn so you can have a glance of what I've been doing, and let's chat there, exchange ideas, and make everything better, right? So the last thing before we start our presentation is my most recent publication. Please check out at the archive 10 quick tips for harnessing the power of ChatGPT and GPT-4 in computational biology. I've made it with a lot of friends like Thiago Lubiana, Rafael Lopes, Juan Carlos Silva, André Nicolau, Vinicius Maracajá, e Elder Nakaya in the University of São Paulo. Please check out the preprint in as soon as possible in a peer-reviewed paper. Okay. So let's get to the main point of your presentation now. Okay, we're gonna begin by the basics. And when I say basic, I'm not saying simple, right? What is bioinformatics? When you search the clear definition in the specialized literature, like the +1 computational biology or the PLOS biology, we find a heated discussion about it. You're gonna find my colleagues arguing about it non-stop. They argue about the name, they argue about the area, if the area really exists or not. Some people argument that bioinformatics is only the development of new tools, computational tools to biology data, other that the use of any computational tool in biology defines bioinformatics. But here we're gonna keep it simple as possible. And this means that all biology today is computational biology or bioinformatics, as you may want to say. I'm personally inclined to agree with this publication. Please check out in the reference. Because today, if you want to work in high level, at any high level, with biological data, you have to use computational science tools. There's no way to deal with biology today like Darwin would do, or like Mendel with, say, I don't know, some notes on plants. You have to deal with computational science to deal with biology today. So all biology is computational biology. If you're the one that needs a formal definition, here it is, and let's make it as simple as possible. Bioinformatics combines computational science and biological data in order to study biological systems. As general as it gets is the way we are going to use today, okay? If you really want to dive into the controversy along the term, please check the references, but here today we're gonna be really simple, okay? Okay, so now that we know what is bioinformatics, let's go to why use bioinformatics in our studies and how it impacts the pharmaceutical science. Okay, I brought these two images to show clearly the why. These are two of the most important resources containing databases of biological data in the world, the GenBank from NCBI and the Interpro from EMBL. NCBI GenBank in 2019 had 7 million genetic code sequences. I'm talking about whole genomes or small sequence, but a lot of them, 7 millions. I don't have the actual number today in 2013, 20, I'm 10 years old on this number, but the point is, it's a lot of sequence, much more than the human could deal without machines. Interpro is the same thing. It houses protein sequences in the same number, thousands and thousands of families, each family with thousands of proteins, each protein with thousands of sequences. There's no enough carbon, energy, time, people, and paper in the planet to deal with it reasonably without the use of the information technology. So it's mandatory for professionals like me, like you, that deal with biological data, such as pharmacists, to use computational tools not only to access this data because it's really far, it's not a physical library, it's a server in some place, not only to access but to extract meaning from it. That's the real important thing. Meaning, we need these computational tools to extract biological meaning in order to make our research, right? So, in simple terms, it's too big to deal with it alone, with our hands and brains alone. This data needs to be addressed with computational science. Aside from the pure mass of data, the techniques, the new techniques that are being developed to extract meaning also needs computational, like quantum calculations, simulations. We need big computers to deal with it. More and more, we deal in silico before we can work in vivo safely, right? This has a huge impact in the way we pharmacists work, especially in safety and ethics concerning biological data in pharmaceutical sciences, right? So, in practical terms, how this will impact your day by day in research, along with the classical tools like the ones in the lab, like PCR machines and microscopics and pipettes and fluid things, we have other tools to deal with this data. Here I choose some of the most common ones like R, Python, the NCBI database, Bash. But if you don't know any of this, if you've never seen any of this, take notes for at least one name and one figure. The small penguin on the lower right side is called Linux. Most part of the development in bioinformatics is made in the Linux environment, an open source operational system that is really, really powerful and basically is the most common ground between bioinformatics and computational science. So if you come to me and say, hey, Pedro, where should I begin? I know nothing. Begin with Linux. If you are a professional with biological background and want to learn bioinformatics or any part of it, begin with Linux. This is the common ground for everyone. Okay? And actually, these things are not really separated like this. They are not apart. You know, most part of professionals deal with any of this, with the both of these kind of tools. We always take new data from PCR and microscopics and everything, and deal with the data with computational tools. Okay, so there is not separate professionals here. You need both, right? Okay. So now I hope that I made it really clear what is bioinformatics and why use bioinformatics in general terms. And I hope that we have a solid ground to discuss upon it. But now I will bring some practical examples in the pharmaceutical science, mainly in research and development, where the professionals use bioinformatics. And the bioinformatics really impacts the day by day in the industry and academy, right? The first one is about drug development. The use of bioinformatics in the process of discovering and making new candidates of drugs to treat disease, right? I will make some citations of specific tools and databases, but please take it with a pinch of salt since the area is evolving in a really, really, really fast pace. And this could change literally tomorrow, right? Okay. Um, so this is a non-exhaustive list of processes regarding drug development that are present in a normal drug discovery pipeline. We could summarize everything here in just one term: molecular simulation. What can I know about a molecule in a biological process before dealing with it in the wet lab? Can I make some observations of expected behavior based on previous data? Can I generate in a cheaper, simpler, faster way in the computational environment before trying it in a chemical biological environment? These are the questions that this process and with molecular simulation intend to answer. And I'm gonna talk a little bit about two specific process and tools actually. The first one, molecular docking software, software that deals with the interaction between a molecule, in general a protein or an enzyme, and small molecules, in general drugs that we develop to interfere in this relation, right? And the second one that I'm gonna explain here is pathway analysis tools. How a pathway in general in the metabolism of some source of some kind in an organism can be accessed through some tools in bioinformatics. Okay, so let's check out. First of all, I'm gonna talk about Autodoc. If you deals with uh, drug design or molecular simulation, you must know about Autodoc. Autodoc is an open source free tool. What it means, the main code is open to anyone to see and tinker with it. Use it to analyze molecules, especially the protein ligand dynamics, which is basically the main part of pharmaceutical sciences, right? We deal with big molecules with small molecules. With this free software, I can simulate an astronomical number of combinations between proteins and small molecules, checking if they are more or less optimal to be tested in vitro. So I can test for a lot of known data, free known data on database for a lot of combinations between possible drugs before going to the lab. What this means in practical terms? In practical terms, these combinations will give me some leads to make in the real world, the material world, the tests in a cheaper way before choosing the ones that really matter to my specific interest. For example, I won't deal with molecules that don't have the characteristics that I want to. And I'm gonna access these characteristics in the computational environment, right? When I go to the bench work, I'll have only the most promising molecules that I accessed in Autodoc. Let's see a little bit how I can make it in the program. So here I have two images that I took out of Autodoc, and here we can see that I can simulate a whole molecule in yellow, and I can see specifically the pockets, the enzymatic pockets of this molecule. In these pockets, I can have a lot of different informations like volume, like charge, like pH, a lot of things. And I can test different molecules in these pockets. I can see the interaction of these molecules. In summary, I can give myself a nice prospect on what to expect of a molecule before using it in the real life, right? Uh, and you can ask me, Pedro, is it perfect? I can, for example, substitute the real test for it, and I will say no. Does it eliminate the need for in vitro and in vivo tests? Absolutely not. But it can save a lot of money, a lot of time, making it safer and cheaper before I go to the actual tests. Okay? So I can have a little glance of it before I go into expose me in the lab to a unknown molecule, right? Okay, so let's get to the next tool. It's called KEG, is the Kyoto Encyclopedia of Genes and Genomes, and it's not only a tool but a database with several different biological data where I can have a visualization of pathways and infer where to interfere, giving a clear clinical objective. Let's visualize it so we can have a clear vision of what I'm talking about. And this is the P53 signaling pathway that I took out of CAG. You can access them the same way in the internet, like in CAG. So access CAG and search for this P53 signaling pathway. And as you can see, I have a vision of several different proteins in the signaling pathway. Not only proteins, but processes, right? I can understand how this pathway works in a healthy individual and infer the impact of an interference in any protein along the way, right? So let's think for a minute. If I want to develop a new drug regarding any protein in this process that I'm seeing, I can have a nice overview of some possible consequences regarding it, right? As I mentioned in the molecular simulation, it has the potential to improve cost saving, safety, and assertiveness in the process. I have a lot of previous data that other people research for me, and I have access here to improve my own research and put my efforts on what's unknown, right? So let's summarize what we've seen until now. Using these tools, I have several different options in the research. If I want to develop a new drug, I can go from a molecule, I can go from a target, I can go from a pathway. I can choose a lot of different things in these databases and tools. And I can have a nice view of the system before going to experimenting it on a chemical or biological way. So I can check everything is known about it and concentrate my resources in what's important, the research, the unknown, my question, my hypothesis, right? Okay, I hope that this was clear. So we can go to the next practical examples on the other side of the pharmaceutical profession, right? I'm going to show more the patients and application side. How bioinformatics can impact not only the drugs, but the clinical perspective of its use, right? How this huge amount of data and accessibility will change the way we deal with patients, with clinical trials, with pharmacovigilance, and so on, right? So to begin, I will be ultimate simple, right? The main difference is that now with bioinformatics, we can address much more details and differences in groups and individual level, right? So we can not only choose an individual based on superficial data like age, ethnical group, and so on, but we can access genomic characteristics, mainly the ones with correlation with clinical relevance to a given objective. For example, if I take COVID-19 pandemic, or for example, here in Brazil, the fatal microcephaly that followed chikungunya infections in pregnant human, we already have studies dealing with the genetic difference between people that will be exposed to the same conditions, but will develop the worst case and will not develop the worst case. This is the kind of difference that I can have access with bioinformatics, right? And here again, I made a non-exhaustive list of impacted areas, and I choose some to illustrate using a nature study from 2018 on the next slide to illustrate all these points and how we could use bioinformatics in our benefits to research about them. So what is this? This is a GWAS study. Pedro, what is a GWAS study? GWAS stands for Genomic Wide Association Study. In this kind of study, generally, we characterize a whole population for a phenotype, like here, kidney stone, and observe with genetic variants which of these genetic variants have association with this phenotype in this population. So here, as you can see in the graph, we have like a dozen of variants highly associated with manifestation of kidney stones and so on. Does it mean that these genetic variations are the cause of the disease? No, I'm not saying that. Does it mean that these variations are really related somehow to the disease? Probably. But it's an association study. More research is necessary to improve the causability, but even with the boundaries of these studies, we now have more information on the genetic background of individuals in a given population and for a given condition. So you can deal with it more specifically and research more about these variants to see if there's a causation relationship, right? So with in the base of genetic background, I can have hints about several different variables of pharmaceutical importance. We know that as a pharmacist, there's a huge difference between individuals regarding the application of drugs in a given disease. Some drugs work better in some individuals, some are more toxic in some individuals, and you know that this has something to do with genetics. If I can have the information regarding the variables, the variants, the genetic variants that have relation with these variables, I can better switch treatment for a particular individual or population, right? More than that, more than treatments, I can think about the prevention side of it. This kind of information have the potential to give personalized advice for people based on their genetic background. So this has a lot of implications in the industries, in performance in sports, in well-being, in insurance, and so on. Think a little bit about it. If I have the genetic background of a people and I know that this genetic background is correlated with a higher risk of a given disease, I can give the specific person personalized advice in order to prevent this disease. I can have it not only for an individual but for entire populations, right? So bioinformatics really improve our actions here. Okay, imagine last example. If I'm going to do a clinical trial, and I have in clinical trials a lot of dynamics in randomizations to make it statistical relevant and so on. I can have this genetical information as variables in these studies. It's not only how the age or the ethnical background. I can have which genetical variants does this population have, so I can use it in my benefit. Okay. Okay. So I hope that these examples show a little bit of the big picture of bioinformatics. And on this third part of our conversation, I'd like to present some innovations and emerging trends that are really hot, that are boiling right now. Okay, I'm gonna do it in a fast way. And the first one is obvious, the hype over AI. And the most famous AI in our area is AlphaFold. AlphaFold functions in a way that you can pretty much simulate any protein with only its sequence. It's not perfect, but it's good enough. And this means that this good enough makes a real revolution in our area. Not only the AlphaFold, but multi-omics. We don't have only the genomic side, but we have proteomic side, we have lipidomic side, and we can integrate all the information, giving us a much more complete map of chemical information in our body, not on individual level, but in population level also. The CRISPR-Cas9, it's not bioinformatics CRISPR-Cas9, it's wet lab. But now, with this technology, the gap between the simulations on computer and the real world modifications on an organism are really narrow. I can edit this DNA in a computer and make the edition in real life really fast in a precision manner. Okay. And last but not least, I think that one of the most important tools that are being developed now that have a relation with bioinformatics is 3D cell culture and organ-on-chip. What this means in practical ways? This means for the biology what the miniaturization of electronics meant for computers in the past. So we are developing machines that can handle thousands of tasks on the same time without the need of big reactors, big volumes, test tubes, and so on. I'm talking about experiments using nanoliters, for example, so I can test several different conditions in the same time, giving me much more velocity on the research, right? This picture here is a summary of everything I've been talking about until now. It illustrates everything. It's from a 2018 paper in Nature, and here you can see a pipeline with several different tasks, in silico to in vitro to in vivo. And it's deeply automated, not only in the computational process, but also in the vivo process, right? And even in the decision-making process, we can have automation. This is a good summary of what we saw now, and it's because we have to the access to the biological data, the processing of this data, the conversion from pure data to organic data mediated by machines, and even the decision-making tasks of this experiment is being made automatically by the algorithm, refeeding the cycle until a person steps on and make it stop, right? The human part here, so they can concentrate on the interpretation of the results and decision making on a higher level. I don't have to deal with the lower level decisions, right? Imagine how many employees in a lab would be necessary to make it by hand, right? So in summary, this picture represents a good chunk of the present potential of bioinformatics in pharmaceutical science, at least in my view, right? Okay, so we've talked a lot about technology here, but I think that we, as professionals, technicians, scientists, students, we've made a very good job with the technical part until now. I am absolutely not afraid that this development will fail in the future. I'm far more concerned about the externalities of these technologies. Here we have a good overlap with the social media and the information technology problems. These data, biological patient data, can literally identify you. I can have a lot of personal information from it, and based on this information, I can take decisions. So, for example, imagine that I'm a politics, or I am a CEO from a biotech company that have these informations. I can take decisions that will deeply impact you based on your biological data. So it's important to define boundaries in this process. Who owns this data? Who can use it? And for what purposes? This is absolutely not clear now, right? I'm gonna give you really extreme examples, but think about for a moment if you are a bad guy. And if you can make a personalized drug, a personalized medicine, I can also make a personalized poison, for example. It's possible. It's possible. Without ethical concerns, it's really possible, really easy. I can really be eugenics about it, right? It's possible. So, in my view, the most important side now, the most important challenge now, goes around this ethical and political part, not the technical part, right? These are some general directions that need to be urgently addressed by professionals like us with deep roots not only on the biological and in the computational side, but deep roots in humanities. I'm talking about philosophy, I'm talking about history, human rights, sociology, diplomacy, these kind of things. We need professionals and funding, not only professionals, but funding focused on ethics and in order to make it real in the real politic, right? In order to guarantee the fair use of these data for the best of objectives of humanity, right? So, in conclusion, given all the advancement and automation, the decision-making process that we need to do will guarantee the need of a good professional here. The robots will not substitute us. Don't make me wrong. The robots today can take decisions for us. They can, but they can't be accountable for it. They have no responsibility at all around it. You need a professional pharmaceutical to be responsible for this process. So if you want to be relevant in the future, um, don't focus too much on the tools specifically, but on the process. Because a new tool will come. Today is bioinformatics. Tomorrow, I don't know, it's other approach. But the pharmaceutical science, the process will prevail. And real people will be needed to guide these tools in this process around it, right? As I said, the main deficit and opportunity that I see as a professional here is around the difficult questions concerning the best ways to use these technologies, ethics, sustainability, accountability, and so on, right? Um, but but if you are really interested in technology development like me, if your heart beats in the rhythm of the zero and ones of a binary machine, be welcome. Be welcome and be prepared because this area is really fast paced and there's a lot of room to make nice and cool stuff. Today I'm programming Python, R, and using a lot of different programming languages, but I don't know if tomorrow there will be an AI that will make all of it obsolete. So you really need to be studying every day, every time about what's going on, right? If you know the metaphor from the books of Alice, like Alice in the Wonderland and something, you know what I'm talking about here, right? So I think that this is it. Here's my contact again. Please check out in LinkedIn and check out the academic reference about it. And I hope that now you have a different and clear view of the impact of bioinformatics in pharmaceutical science. Thank you very much and see you there.